Yeah, good for them.
How's the girl doing this morning?
What's that?
How's the girl doing this morning?
Yeah. 24 hours.
It's like it wasn't working anyway. I think there's something wrong with that battery. Well, I mean, like the it wasn't sound. It's not supposed to be that. There is a battery in there that was fresh yesterday, but it's not. Oh wait, it's working now. Wait, hello. Okay, I think it's working now.
So he's just like disappeared, like he might still be present or
Okay.
it's a big room.
All right, everybody, thank you for coming so early this morning. It's really my great pleasure to introduce to you Kimberly Mighell. Today she's going to be talking about microbial symbiosis, perspectives from leaf-cutting ants and Dissertation. But before we get started, I just want to talk about a little bit of Kim's past. So let's see. Kim grew up in Shreveport, Louisiana. And she got her bachelor's degree at the University of North Texas. She got a BS in biological sciences, and she began her research career there. And this included an opportunity, an NSF-funded opportunity to go to Chile and do some research there. And after graduating, she did an internship at the Field Museum of Natural History in Chicago, which is funny because I had an internship there too when I was an undergraduate. She started as a graduate student at Tulane in 2012. And let's see, I gotta wait for it back up. And Kim was my very first PhD student, so she helped out a lot with modeling new lab spaces. You can see behind Kim and myself and Demetra that there's nothing in the lab. Pretty much all we had was a printer. And she really took it seriously, modeling these new spaces. Those are some jatropha plants behind Kim there, and the breezes of the growing chamber blowing her hair back. She started research on leaf-cutting ants in Panama. She was willing to tackle the jungles of Panama and all of the great predators there. She was really great at mentoring undergraduates. So here you see some little kids, that's me and my kids. We're digging up some leafcutter ants, and Kim was helping to mentor two undergraduates that you see here. And there were lots of other undergraduates, you'll recognize some more faces that she helped to mentor along the way, and some more that she tried to keep out of trouble. And Kim's always been great with kids. These are some of the lab kids and some of the lab grandkids. Besides that, she's been really great at helping to mentor girls through our Girls in STEM program. She's always been a big supporter of that program. So we have always known that Kim can also be a little bit silly. This picture was taken just this week. And Kim is part of a Mermaids on Unicorns Cavalry marching through the streets for Mardi Gras. That's her. It's hard to see the mermaid tail, but it's there if you look closely. So Kim's always been a wonderful part of the lab. She has guided everyone through, and I'm speaking for all the lab to say that we're really proud of her, and we're going to miss her a lot. And now she's going to talk to you about her research. There we go.
Well, thanks, Sunshine. That was really lovely. I had no idea you had so many photos of me.
I have help.
So it's been a real privilege to be at Tulane for the past 5 years, and I'm looking forward to sharing my dissertation work with you all today. What I've been doing these past 5 years is overall looking at microbial symbioses in a lot of different contexts. And today I'm going to talk about microbial symbioses in the leaf-cutting ant system and with Dissertation. So traditionally in ecology, we treat organisms as discrete entities with one unchanging genome. However, the field of microbial ecology, can everyone hear me okay? This is good? Okay, I'm kind of loud. The field of microbial ecology is teaching us to treat macroorganisms as holobions. The holobiont concept is the organism, the macroorganism with its genome and its collective microbiota, so the microbiome here. The microbiome being all the bacteria and fungi that reside inside of the organism and on the surface of the organism. The composition of the microbiome could change over space and over time, and it interacts really intimately with its host. So the microbiome can directly alter host phenotype. It has genetic elements which can be tightly co-evolved with host genes to work together to influence host phenotype. But it has a lot of unknown functions, so there's a lot of not obvious or neutral effects of the microbiome on the holobiont. And finally, we have to remember that the holobiont lives in a microbial world. So there are microbes all around the holobiont that maybe, while not directly interacting with the microbiome, could affect its interactions with other organisms. So why are microbes such good partners? Why do we think about microbial symbioses in the holobiont? So while a host only has one genome, the microbiome can hold thousands, and this can facilitate novel strategies within the holobiont, such as nutrient acquisition. Here we see a seedling which is associating with AMF, or a mycorrhizal fungi. The mycorrhiza is a fungus that interacts intimately with plant roots. It forms an interface with the roots where it receives sugars from the plant host. It grows out into the substrate where it collects trace minerals and nutrients, which it brings to its plant host. We also see microbial symbioses affecting tolerances of holobionts. So a really great example is a grass in Yellowstone, and this grass has adapted to grow in this geothermally warm soils due to the association of a fungal endophyte virus complex. So this grass has radiated its niche into novel territory through a microbial symbiosis. Microbes are also really great at facilitating novel defense strategies. So here we see a blade of grass, and throughout the grass, this is an epicloe fungal endophyte that's been tagged with GFP. We see it growing in between all of the cells, and what it's doing is it's receiving sugars and leachate from between those cells, but it's producing a really toxic alkaloid. This alkaloid makes the grass really bitter, and it deters herbivores, acting as a defense for its grass host. We also have to remember that the microbiome is not always mutualistic. So the microbiome operates on a mutualist pathogen continuum. If the microbiome parallels its host fitness, then we think of it as a mutualist, where the microbiome benefits and the host benefits. But it can slip down into parasitic, say if the burden of hosting a very diverse microbiota is expensive and the host becomes stressed, the cost of that microbiome might become parasitic to that host. Also, if there is a disturbance in the microbiome community, certain pathogenic elements of that community may take advantage of the disturbance, gain in abundance, and bring that whole the microbiota fitness down towards parasitism. So it's really important to remember that microbes are good partners, but they also can be bad partners in certain contexts. So by using the holobiont, we can develop a couple of novel questions. First of all, what determines the composition of the microbiome? Is it those microbes that are in the environment? Are there abiotic influences? Or is there some degree of host selection in selecting for certain members? Secondly, what effect does the microbiome have on the holobiont? So where in time, or where in space on that mutualist pathogen continuum do we find our microbiome of interest? So we're going to visualize these questions through a schematic. If this is our holobiont, within the holobiont is the microbiome. The microbiome and the holobiont can have positive and negative effects on each other. And that microbiome, in large part, is derived from the environmental microbes. So things in the environment that can colonize the host to develop the holobiont. This microbial species pool, as it's recruited into the microbiome, is constricted by abiotic factors. These are things like pH, chemistry, things that would affect the metabolism of microbes in the environment, favoring certain members and disfavoring others. And these abiotic factors can also stress our holobiont. Biotic factors in the environment also play a role. So other biotic members in the local community could alter the metabolism and favor certain species through making certain sugars or more bioavailable, which would favor certain members, or maybe by directly competing with other microbes in the environment. The holobiont could also locally manipulate its space by providing certain sugars or secreting certain enzymes, which would favor or discern certain endosymbiotic groups from its local environment. Of course, these biotic factors can also stress the holobiont if they're directly competing with the holobiont, or they could be diseases. So today I'm going to show that leaf-cutting ants and Jatropha are engaged in many different overlapping microbial symbioses, and that these symbioses range from mutualistic to pathogenic. I'm going to do this in 3 chapters. In chapter 1, we're going to look at leaf-cutting ants and microfungi. In chapter 2, we'll look at Jatrophococcus and endosymbiotic bacteria. And in chapter 3, we'll look at Jatrophococcus and the soil microbiota. So the leaf-cutting ant system is a really good system to look at microbial symbioses because there's a lot of partners that overlap in their functions here. So the leaf-cutting ants, these are the attine ants. They're not found here, but they're really common in the tropics, and we do find them in drier parts of Louisiana. They actively cut vegetative material when they're out foraging, and they bring it underground, and they use that vegetative material as a compost with which they grow a fungal garden. This fungal garden is a monoculture of one Leucoagaricus fungal species, but within that garden, there exists many other bacteria, yeasts, and microfungi which are not obvious to the naked eye. So the ants are the fungal garden provides a reliable year-round food source for the ant colony that they wholly depend on. And the ants are solely responsible for caring for that garden, for bringing it food, and for keeping it healthy by pruning it and by controlling disease within the garden. I should note that this fungal garden is only found in association with the leaf-cutting ants. It's not found naturally, or it's not found outside of this mutualism, rather. The leaf-cutting ants also interact with other fungi in the environment through the leaves that they cut. So almost all leaves that they cut contain endophytic fungi, which are the fungi that exist within leaves with no obvious symptoms. They reduce loads of the endophytic fungi before they plant them in their garden through application of antimicrobial secretions to clean the leaf piece, but also through selecting leaf substrate that's lower in endophytic levels. These endophytic fungi can directly compete with the fungal garden, the Leucoagaricus, for that leaf substrate, and in turn, the fungal garden has some degree of antibiosis against those endophytic fungi. Leaf-cutting ants also interact with a lot of soil and parasitic fungi in their environment. Remember, their colonies are underground, and the soil is full of fungi and bacteria. These soil and parasitic fungi, the leaf-cutting ants can reduce loads of these in their garden through actions of pruning and weeding and spot treatment of their fungal garden with antimicrobial secretions. So they're constantly going through their garden, looking for any places that are sick, and removing that fungi. The soil and parasitic fungus can have a negative impact on the fungal garden if it can establish. So it can act as an active mycopathogen, so directly parasitizing the fungal garden, or it can act as an opportunistic antagonist, sort of just a saprotroph, sort of just like cheating and leaching off all the nutrients in there. And the garden, again, has some antibiosis capabilities against these soil and parasitic fungi. So this leads us to our motivating question, which is leaf cutting ant behavior towards all external microfungi general or specific? And we approach this question by utilizing bioassays of miniature leaf cutting ant colonies and challenging them with different cultures of microfungi. So we did this work in Gamboa, Panama. We went out into the forest and we found leaf cutting ants that were actively foraging and bringing vegetative material into the nest. We collected those leaf pieces and we generated a endophyte culture colony from those. We then went and we dug out some fresh nests. We plated their garden fungi on agar, and in the absence of the ants, all of those pathogens and opportunistic antagonists will grow out of the Leucoagaricus fungal garden, and then we can isolate them in pure culture. So we had 2 culture collections of endophytes and garden contaminants. We also had a strain of contaminant that we isolated from a dead leaf cutting ant within our lab colony. So we generated these microcolonies. These were really cool. It took us a long time to get this set up and working. So from one active leaf cutting ant colony, we made these little microcolonies in 4-chambered petri dishes where we had a water source. We had a scoop of the fungal garden, the Leucoagaricus, that also contains larvae, media workers, and minor workers. We had a scoop of their trash to simulate their naturally, so they would know where to take sick parts of the garden and things that they wanted to remove from the colony. And we had an empty chamber. We cut 3 plugs of fungus from each fungal culture strain, and we had 3 control plugs of just agar. We introduced each of those plugs into a microcolony, and after 3 hours, we calculated the effect of the ants by measuring the percent of the fungal plug that was removed, or the control plug that was removed. And we calculated the effect of the ants by using Glass's delta. So what did we find? We actually found no difference based on isolation source. So all of our endophytes and all of our contaminants did not have significant differences on their removal rates. But within each group, we see a big range of reactions. So with our endophytes, about half of them had really high response rates by the ants, and about half of them were sort of left alone by the ants. And we see the same thing with our garden contaminants. And we can characterize about half of these as parasites based on the literature, and we can characterize about half of these as saprotrophs based on the literature. And so why the variation within these groups? So we know that these parasites, Escovopsis, Trichoderma, and Paleomyces, can be very destructive to leaf-cutting ant colonies by parasitizing the fungal garden or the ants themselves. Saprotrophs, the 3 saprotrophs that we found, have really no known mycopathogenic or entomopathogenic capabilities. So the fact that they weren't removed in high rates by the ants isn't really surprising. But the endophytes are actually really neat. So in the forest, almost all the leaves have a high diversity of endophytes, and 80% of these endophytes produce biologically active secondary metabolites. So this means that these secondary metabolites could have fungicidal or insecticidal or antimicrobial capabilities. They could alter the taste of the tissue that they grow on. And we've also seen in lab colonies that ant colonies fed a diet high in endophytes grow slower than control colonies. So there is probably some sort of effect of these biologically active secondary metabolites in the fungal garden. So going back to our original question of is leaf-cutting ant behavior general or specific towards all these microfungi, we can pretty much safely say that it's specific, and that the ants are able to discern strain-level differences of the fungi and modulate their behavior. And this behavior modulation could provide some insight into the ecology of these endophytes within the leaf-cutting ant system. So going back to our schematic, oh, that's sort of blue. Okay, well, we see that if the holobiont is our fungal garden, and the microbiome of the fungal garden is right here, then our ants act as biotic factors in the system to restrict the diversity of microbes that are entering into the holobiont. So they're actively discerning different fungal strains. Okay, now we're going to shift gears a little bit, and I'm going to drink some tea. So I want to talk about Jatrophococcus with y'all now. This is what I did I used as a model system for my second and third chapters. Jatropha is a big woody shrub. It's in the Euphorbiaceae family, which is notoriously chemically complex. It produces a lot of antimicrobials and toxins like forbolesters and cursins within the tissue, which make it really nasty. And it's also used as a traditional medicine because of these biologically important properties of the plant. However, a lot of people are interested in it because it's a very useful biodiesel. It's grown in the tropics all over the world as a biodiesel crop. It produces these big, oily seeds, which, when they're pressed, can combust as a diesel. And it's grown in plantations around the world, but in Panama, we most often see it growing as a living fence. So here, farmers, this is Jatropha growing here. Farmers use it as a sort of a hedgerow to delineate properties because the cattle don't eat it because it tastes nasty and it's full of toxins. It has these big leaves that provide a lot of shade, and it's very resilient. It's very hardy. So Jatropha grows, it's very drought resistant. It can survive the dry seasons, and it grows in a lot of different soil types. And because it's so chemically complex, because it's so drought resistant, because it grows in a lot of different soil types all over the world, we propose that it's actually a really good model to study how plant microbiomes assemble under very different conditions, and then to see what effects those microbiomes might have in Jatropha. So a little bit about plant microbiomes. So plants and soil are very important. As the plant, well, the green doesn't show up around my rhizosphere, but as the plant roots grow out into the soil, it forms this really intimate association around the roots known as the rhizosphere. This is this zone surrounding the root tissue. Upwards of 40% of the carbon within roots can leach out into the rhizosphere. And so the rhizosphere is enriched in sugars, but it's also enriched in polysaccharides and ectoenzymes that the plant produces that exude out into the rhizosphere. This signature of exudates can be species-specific, or in some cases, even genotype-specific. And because there's this really interesting sugar-rich chemical signature in the rhizosphere, it's attracting a lot of soil symbionts. So be those pathogens that are looking for a specific host, it can find that host through chemicals in the rhizosphere, or be those mutualists who are looking for a specific host, it can find that host through the rhizosphere, or be it just bacteria and fungi that are looking for some quick sugars and they're attracted to these sugars within the rhizosphere. So the rhizosphere is a region which is enriched in microbes, and it's usually a composition of microbes which is distinct from the bulk soil. From the rhizosphere, the endophytic microbiome is recruited. So we see bacteria and fungi which enter in between the cells of the roots to colonize the inner tissue to become the endophytic microbiome, while the fungi will mostly remain localized within the cortex of that tissue. Bacteria can actually travel in between the cells into the inner root, where a subset of the bacteria can enter xylem tissue and then move vertically into the shoot, colonizing shoot tissue. So we see a filtering of the bacterial community from bulk soil to the root tissue, and then a special subset colonizing the shoot tissue. So what do we know about plant microbiomes in Jatropha? Well, despite it being really chemically complex, we've been able to culture bacteria and fungi from root and shoot tissue. So that's good. It's got microbes in there. We also know that Jatropha is able to change the soil microbial community. So Jatropha plantations, after they have been established for several years, actually have a different soil microbial community than adjacent lands. So probably the root exudates are pretty important. We also know that Jatropha hosts AMF. So arbuscular mycorrhizal fungi are a subset of mycorrhizae. They form intimate associations where they actually penetrate the host cell wall and they form an interface with the plant plasma membrane. Across this interface, they absorb sugars from the plant. They grow out through the rhizosphere into the bulk soil to get trace elements that they bring to their host. So we can develop a couple questions thinking about Jatropha's microbiome, being what influences Jatropha's endosymbiotic bacterial community, and then secondly, what role did the different soil microbial elements have on Jatropha's traits? So going back to our schematic, thinking about abiotic factors and local microbiota, if Jatropha is our holobiont and its microbiome is right here, most of the microbiome is assembled from the soil. So the soil serves as our microbial species pool. Within the soil, abiotic factors can directly constrict that diversity. Really strong abiotic factors that explain soil bacterial community are soil adaptive factors like moisture, but also pH, very highly important. Other anions can also explain soil bacterial communities. These abiotic factors can also stress the host if maybe the soil is low in plant nutrients or low in plant moisture, or low in moisture. And this stress of the host can indirectly affect the microbiome. If it changes Jatropha's exudates in the rhizosphere, we would see a different community assembling around there. So the plant could locally manipulate the microbiome within the rhizosphere. Also, if the plant is stressed, its internal chemistry or its immune system might begin acting differently, and it could, within its tissue, interact differently with the microbial symbiotic community. So this leads us to our hypothesis that the endosymbiotic community is affected by both the environment, and when we say environment, we mean the ambient environment, but we also mean the soil chemical environment, as well as the soil microbial community. And we address this hypothesis through both field and greenhouse studies. So we grew plants in 3 field sites, and then in a controlled environment in the greenhouse with soil from those 3 sites. So if soil factors, those being soil chemistry and soil microbial community, alone drive the assembly of the endophytic community, then we would expect to see that plants in the same soil, regardless of ambient environment, would assemble the same endophytic community. However, if ambient environment plays a role, so if things like temperature or soil moisture or just like local conditions affect that, then in the greenhouse, where the environment is controlled and the same for all of our plants, we would expect to see a similar endophytic community assemble within those hosts. So to address this question, we chose 3 sites in Panama. So this is the Azuero Peninsula of Panama. It juts out into the Pacific Ocean. And this region around here is very dry. It's called the Arco Seco, the dry arc. It's where a lot of the cattle country is. Relative to the rest of Panama, it's fairly dry. This didn't show up so great, but we chose a transect along the eastern side of the peninsula. Our northernmost site, Divisa, contains a 3-year-old Jatropha plantation that's fairly rainy. Our central site, El Ejido, contains a 3-year-old very dry Jatropha plantation. And our southern site, Los Asientos, is a fairly rainy cattle pasture where Jatropha is found growing as a living fence. In our 3 sites, we set up 2 plots per site. Each plot was 8 meters by 6 meters and contained 12 subplots. Within each half of those subplots received 1 kilogram of agricultural lime. This agricultural lime physically manipulated the soil to raise the pH by 1 unit. So agricultural lime is just ground up limestone, calcium carbonate. So what it does is it just increases the pH of that soil and introduces calcium. So we had 3 sites, 2 plots per site, 12 plants per plot, for a total of 144 plants. We grew seedlings that were in sterile conditions in Gamboa, And after several weeks, when the limestone had been able to seep into the soil and actually change the chemistry locally, we went out and we transplanted each of our plants into these subplots. And we let them grow for 10 weeks. I monitored them for about 10 days to make sure they all established and to put down water while their root system was developing. But after that, I got out of there and went back to Gamboa. We took soil samples where we measured the pH, the cations, and the total phosphorus of all of our soil pre and post liming, so we could see the effects of the liming. We grew plants in the greenhouse in soil from those 3 locations. It was a 10% soil inoculum to 90% vermiculum to account for maybe any major differences in nutrients that we would see. And finally, we got the DNA. So we extracted the DNA from the soil roots and leaves. We amplified the 16S region, which is the bacterial barcode gene. We sequenced our libraries on a MySeq at NAUS in Panama. And then we did downstream analyses with the Chime pipeline, and we did SAT analyses with the R packages PhyloSeq and Vegan. And first of all, let's talk about the sites. So we see that the rainfall varies between the sites from wet Divisa, dry El wet Los Asientos. Our pH at all the sites in our unlimed plots was very similar, about neutral. In our limed plots, also always raised about 1 pH unit. Yes, it worked. At Divisa, we had really high phosphorus levels. Was not expecting that. This field had been historically fertilized, so we actually had really high levels here relative to our other 2 sites. Our potassium was higher in our 2 plantations than it was in our cattle pasture. Magnesium was really high at El Ejido, and we see calcium pretty much correlating with the application of lime, which is what we would expect since we're just applying grind-up limestone. These are all important to look at because these highly correlate with bacterial community structure in the soil, but also with plant growth rates. So these are important plant primary and secondary nutrients. So this is an ordination of our soil bacterial community structure, principal coordinates analysis. And what you need to know to interpret this graph is that each one of these points represents one bacterial community within one sample. So the closer in space 2 points are, the more similar those bacterial communities are, and the further they are in space, the more dissimilar that bacterial community structure. So what we see first of all is that our 3 sites had significantly different bacterial communities. So across the peninsula we see differences in soil microbiota. Secondly, within each plot, we see a predictable shift along axis 2 due to the application of agricultural lime. So when we applied lime, we could change the soil microbial structure in a predictable manner. Secondly, when we look at the bacterial community structure of our endosymbionts in the field, we see that there is a filtering of diversity from high diversity in the soil, middle diversity in the roots, and lower diversity in the leaves. So that's what we would expect if the bacteria are being recruited from the soil to the roots and then into the leaves. What about the bacterial community structure? So this is an ordination of the bacterial community structure of our leaves, our roots, and our soil. First of all, we see that our community structure is significantly different based on isolate source. So our soils, our roots, and our leaves all have significantly different communities. Within that isolate source, within the roots, we see significant separation of communities based on site location. And we see the same thing in the leaves, maybe not to such a strong degree because the community is more similar, but it's significantly separating out by field location. So going back to our predictive graph, we see that the endophytic communities vary across the field location where we know that the soil chemistry, the rainfall, and the soil microbes are variable. So in our 3 sites, which vary in all of these qualities, we see different endosymbiotic symbiotic communities assembling. What about the greenhouse plants? So looking at the roots of the field to the greenhouse plants, we see first of all that our roots in the field were always more diverse than our roots in the greenhouse. We look at the community structure of our roots in the field and the greenhouse. We see that our roots in the field are more distinct by site, and they are also more dissimilar from each other. Our roots in the greenhouse are more similar to each other, and while we can see trends that they're starting to separate out by site, these trends are not significant. The leaves had a slightly different pattern. So in the greenhouse, our leaves were always as rich as or richer than our leaves in the field, which was interesting. When we look at the community composition of the bacteria in the leaves, we see that our leaves in the field were more similar to each other, but our leaves in the greenhouse were sort of all over the place. While our leaves in the field separate out significantly by location, our leaves in the greenhouse do not significantly separate out by soil origin. So, going back to our predictions, we see that in the fields, we had different endosymbiotic communities when there were different soil microbial communities, different ambient environments, and different soil chemistry conditions. But in the greenhouse, when we just had different soil chemistry and different soil microbial communities, but the same ambient environment, we saw a convergence of communities both in the roots and in the leaves. So something that we want to answer now is which factors play a role in driving these differences that we see in the field. There's a lot of different things that are happening. We know that there's differences in soil chemistry, there's differences in rainfall, and there's differences in the soil microbes. So how can we explain the variance based on all of these different things that we're seeing in the field? So to answer this, we built a model. We did a DBRDA, a distance-based redundancy analysis. And to do this, we put into our analysis our root dissimilarity matrix. So this is the matrix that explains all of the root endosymbiotic communities across all of the field sites. We trained our model on all of the soil chemistry data, the rainfall data, but also the soil microbial data. And how we extracted that data is from our soil ordination. When we looked at the dissimilarities of all of our soil communities across sites, we extracted the principal coordinates axes for each of those sites, and we put those axes into the model to explain the soil microbial data. Is everyone with me? Okay. So we trained the model on all of that data. We let the model select which of all those variables best explains the root endophytic community. And then we selectively constrained the model for the components in the environment that it said was significant and the components of the soil microbial diversity that it picked as being significant. And what we found is that the environmental variables, potassium, phosphorus, magnesium, calcium, aluminum, and rainfall, significantly explain 24% of the variation in the roots. That's a lot. That's exciting. Also, 4 axes of the soil microbial community explain 11% of the variation. So in total, our model was able to explain 33% of the variation within the roots just by using this data that we put in. This is really cool because these, the environmental variables that it selected are highly correlated with plant growth and plant health. It can be very informative on overall predicting plant traits. But also, we see right down here elements of the soil microbial community that are significant. Exactly which members of the soil microbial community are driving those differences and why these lower axes in the principal coordinates analysis were selected is a really good question that we don't have time to address right now. So another thing that I want to add here is that when we applied the agricultural lime in 2 of the plots, we actually saw significant differences in our root community structure in the plots that were unlimed versus the plots that were limed. This is really neat because it means that farmers can physically manipulate the endosymbiotic bacterial community of their crops through really common agricultural practices. Application of lime is something that is really commonly done. It's really easy to do. So in summary, we found that the soil community varies across sites. We found that the endophytic community inside Jatropha is very plastic, and this plasticity is affected by the plant organ, so whether it's the roots or the leaves, the environmental conditions, those being ambient conditions, and soil chemistry, and it's affected by the soil microbial community. We also found that we can change the endosymbiotic community with agricultural lime. And overall, we did see that the environment, the ambient environment, and the soil chemistry has a stronger effect than soil microbial diversity in driving endosymbiont assemblage. So going back to our schematic, what we found is that there is significant contribution of the microbial species pool in determining the assembly of the microbiome, that the abiotic factors, those being ambient environment and soil chemistry, are important in constricting and changing that diversity, and also that at the host level we see local manipulation within tissue of hosts selecting different communities and different tissue sites. So, last chapter, now that we've talked about what's inside the plant, let's talk more about the soil. So soil is a really complex matter. We walk over it every day, but we don't think about it too much. It has a lot of minerals, organic matter, gases, liquids, and of course, our favorite, microbes. So we know that soil influences plants, right? Soil is the source of all the nutrients. It's a source of water, where the roots suck up most of the water. And it's also the source of the vast majority of symbionts, those being fungal and bacterial. But what we don't often think about is how plants influence the soil. So we've already talked about the importance of exudates. Here we can visualize a Jatropha tree growing in the field. As it grows out into the soil, it's going to have this zone of the rhizosphere around that root tissue, where it's going to put exudates out into that tissue, into the bulk soil. And so these exudates within the rhizosphere contain sugars, proteins, enzymes, and they can promote certain symbiotic groups, those being bacteria, which we know can get into the tissue, those being AMF, which we know it associates with, or fungal endophytes, or certain symbiotic groups that contain pathogens. And soil-borne symbionts are really important for plant traits. So we know that soil-borne symbionts can influence growth rates. So nitrogen-fixing bacteria in the soil is a really important source of a lot of plant nitrogen. It turns atmospheric nitrogen into bioavailable forms for uptake by roots. So this is a schematic of rhizosphere bacteria that you don't have to totally understand everything that's happening. But what I want to show you is that there's a lot of overlap between bacterial pathways and plant pathways. So rhizosphere bacteria can make minerals and phosphorus more soluble for root uptake. They can be a major source of iron by complexing irons for roots to uptake. But also, rhizosphere bacteria can directly stimulate plant growth by producing plant hormones, such as auxins. And it can directly alleviate stresses on plant hosts by breaking down stress enzyme precursors. Also, it can influence growth rates for the worse. So, Fusarium is a really common fungal pathogen, and this is a banana stalk which has been totally infected with a soil-borne Fusarium, which is causing the devastation of a lot of the banana plants, Panama disease. You may have heard it called. But soil-borne symbionts are also really important for defense pathways of plants. So, all of our microbes that are existing in the rhizosphere and within the plant tissue have these things on their cell surface called MAMPs. These are microbe-associated molecular patterns. These are things like chemical signatures that microbes have on their surface, like bacteria could have flagellin, or the certain proteins that are found in fungal cell walls. And these MAMPs can attach to cell surface receptors on plant cells, triggering a cascade of MAMP-triggered immunity. Fungi that associate intimately with plant cells as well can release effectors into the plant cell, collectively triggering effector-triggered immunity. These two things are called induced systemic resistance, and they create a cascade, a cascade of upregulation of pathogen resistance proteins. They increase cell wall fortifications in plants, and they can alter phytoalexin productions. These are all really important things. If a plant wants to stay healthy, it wants to fight off pathogens. So pretty much it's just like the plant going to the gym and getting like really beefed up so that if someone wants to come fight it, it'll be strong. But also these microbes in the rhizosphere or within plant tissue can directly inhibit pathogens, be that through production of antimicrobials or through competition for that space within plant tissue. So this has led to our hypothesis that the soil microbial community will aid in Jatropha's growth and defense. We approach this through a greenhouse experiment, aiming to separate out the effects of the different soil microbial elements, predicting that the different elements will have different effects on these characteristics of Jatropha. So what we did is in our northernmost site, Divisa, where our plants are for the most part really healthy, they grow really quickly, we collected soil in sterile buckets from beneath a mature Jatropha tree. We took that soil back to Gamboa and we processed it. About half of it was left raw, and half of it was autoclaved twice to remove all of the microbial components of that soil. Of the raw soil, we took some raw soil and used it as our all diversity inoculum. This contained high diversity of fungi and bacteria. We added fungicide. I think I'm covering this with my finger. We added fungicide to a subset of the raw soil to produce a bacterial diversity inoculum, which had fungi suppressed. We extracted AMF spores from a portion of the live soil. We added it to some sterile soil to produce an AMF inoculum, so only AMF exists within that treatment. We had some sterile soil we used as a low diversity inoculum, and then we added fungicide to a treatment to have a fungicide control to make sure there weren't any effects of the fungicide on plant growth or traits. And what did we find? So we let the plants grow in the greenhouse, and we measured their relative growth rate, which is the rate at which the plants grew. And what we found, first of all, is that the high soil diversity did not necessarily lead to healthier plants. In fact, our all diversity treatments and our bacterial diversity treatments performed the worst consistently. Our AMF treatment had the highest growth rates, and our low diversity treatments and our fungicide controls were right there in the middle. We calculated our growth rate as both wet and dry. Wet, I think it's interesting to look at with Jatropha because it's a big succulent plant. It has a giant fleshy stem, and it uses the stem to resist drought. Dry growth rate is just going to be straight carbon accumulation. So we see these treatments separating out really nicely when we look at the wet growth rate, but it's a little messy when we look at the dry growth rate, with our AMF not actually falling out as significantly different from our low or our fungicide controls. What about defense? So maybe there's something, maybe the soil microbes affect plant defense pathways. So we grew Calinectria in fungal culture. This is a generalist foliar necrotrophic pathogen. It's found with a worldwide distribution. We had Calinectria plugs, and we had control auger plugs, and we attached one of each plug to a Jatropha plant in the greenhouse. We let that fungus infect our plant for 3 days. Then we detached the leaf, and we measured fungal lesion size and control lesion size. And what we found is that there's really no influence of soil microbial diversity on Jatropha's defense pathways. So we found that our Calinectria lesions were always larger than our control lesions. That was good. We were able to infect our plants, and our controls did not cause disease. But there was no influence of the soil microbial community on our Calinectria lesions or the control lesions. So we can conclude that a diverse soil microbial community actually negatively influences Jatropha growth. Likely there's some fungal pathogens in the soil that are playing a role here that's decreasing the growth relative to the sterile soil. We may be seeing some possible negative density dependence. So we collected the soil from beneath a mature Jatropha tree, and this soil could be enriched in Jatropha-specific pathogens. If you stick around for 11, I think Scott will maybe tell you a little bit more about what this means. And we saw that AMF does have some positive benefits on Jatropha's growth. But there's really no impact of the soil microbiota on Jatropha's defense against general foliar fungal pathogens. So it could be that Jatropha's induced systemic resistance is just less active, or maybe Counter-G is too strong of a pathogen. We use it because it grows really well in culture and infects things really consistently, and maybe this was actually a bad idea. But Jatropha could be more reliant on innate defenses. So we know it has really complex leaf chemistry, and Jatropha could rely more on this complex leaf chemistry to defend itself than induced defenses. So, going back to our schematic, looking within Jatropha, we see that Jatropha and its microbial symbiotic community have actually, the microbial symbiotic community in the soil can have negative influences on Jatropha, or neutral influences on Jatropha, or positive influences on Jatropha, depending on which elements you're looking at. So going back to our novel questions that we posed in the beginning, can we answer them? Well, what determines the microbiome of the holobiont? We found that abiotic influences are really important, that soil chemistry in the ambient environment are important in determining the soil the endophytic microbial community of Jatropha. We found that environmental microbes are also really important in driving the assembly. We found that there is some element of host selection at the tissue level. And we found that there are biotic influences in the holobiont system, which can affect the assembly of microbial communities. When we go back and remember those ants that we looked at. Also, what effect does the microbiome have on the holobiont? We found that it has positive, negative, and neutral effects. So I hope today that I've been able to convince you to not look at organisms as discrete beings, but as holobionts, and to take into account their microbiome and all the environmental microbes that they could be interacting with. And to remember that these overlapping symbioses have important consequences for host ecology. And with that, I will go ahead and wrap things up. I have so many people to thank. I was here for 5 years, and I couldn't have got through it without a really strong team. So I want to thank my committee first. Sunshine has always been so wonderful. I was her first grad student. We just sort of figured this all out together, and she always encouraged me to follow my nose and go and pursue questions that I was interested in. Scott has been a great support system in Panama and helping me sort of figure out my ideas, and I'm glad that he's here today. Liz Derryberry and her writing group kept me on track and was able to turn my findings into solid chapters. Emily Farr, the latest addition to our committee, helped me figure out my DBRDA and figure out how to partition all my variances. And past committee members, Steve Darwin and Corey Richard Zawacki, were great at setting me on my journey. In Panama, this work was possible due to a short-term fellowship and a couple internships at Stry. My mentors there have been Kristen Saltonstall, Ben Turner, Alan Harry, and Anith Rojas have been really great at helping me get the logistics of everything done. And in the Azuero, Jaime Espinosa and Odiel Solis and Jacob Slusher at IDIAP and LT were really important for setting up all my field sites and keeping my plants alive while they were out there. And of course, this really great support system, all of my lab members, Peter, Liz, Steve, Jen, Emma, Kyle, and Anna, who've helped out in Panama, and Alex, who's helped me do some really monotonous measurements in the lab. Not to mention Creep. Thanks, guys. Y'all been really good, keeping me safe and keeping me fed. With that, also funding. So all of this work was possible because I had a 2-year EPA STAR fellowship. It was amazing. They gave me money to do my sequencing and for traveling. This is a really wonderful program that I hope call your senators and don't let them axe this. This is a really important fellowship. Short-term fellowships and funding from STRI were also really important. And the Stone Center Latin American Studies program helped fund one of my field seasons. And with that, I will take any questions. Yeah, Jordan.
Great job starting off the parade of thesis defenses that we're going to have this semester.
First pancake.
My question is about your first chapter with the ants. And I was just wondering, did you source all those ants from the same colony or from different colonies? And did you look at, if you did do the latter, did you see any inter-colony defenses or would you expect differences?
Yeah, so we had one freshly collected colony per fungal strain. So the day before I would do an assay, I would go and collect a colony, bring them back to the lab, let them reassemble, and then use those. So we used all colonies that were 1 to 3 years old in the same area in Gamboa. So they would have been interacting with very similar microbes in their environment. And we operated under the assumption that all of these colonies, since they're in the same environment and the same age, would respond to microfungi in the same way.
So you mentioned like, farmers can alter the microbiome through simple agricultural process like putting lime. Why would they? I mean, I see benefits of adding lime, but like, why would they do that to alter the microbiome? I'm just asking because, like, in the end, you saw like some positive effects, negative effects, neutral effects. So, what's the guarantee that they would be altering the microbiome in a positive way?
Yeah, so a lot of people are interested in manipulating either the soil microbiome or the endophytic microbiome to enrich in certain groups that they know are plant growth promoters. But also, certain soils that are more diverse or have certain levels of diversity have been shown to suppress fungal diseases in the soil. So if, just by looking at the community composition, we can't say whether the lime enriched the soil in disease suppressors or not. That's something that I'm planning on going back to the raw data and looking for individual families of bacteria to answer those questions. But by looking more in depth at the data to what changed within those communities, you could see whether it would have a predictable positive, negative, or neutral effect on that host. Anyone else? Okay, so I'll see you all at 5.
Yeah. So there will be a social hour in Audubon Park at 5. That's what she's referring to, and you're all invited. Please do come. We'll be celebrating.
Thanks, guys.